Yeah, thanks very much, Tilo, for the introduction. Thank you to all the organizers for giving me this opportunity to talk about this joint work together with with Tilo and also with Valentin, who's working at the EPFL in Switzerland. And yeah, I'm talking mainly about a mesoscopic mean field model for spiking neural networks and how metastability arises in these kind of networks. As we've heard before by Maurizio, metastability, or one of these important or key factors for metastability, is some activity-dependent fatigue mechanism. This can be either like via spike frequency adaptation, mediated by calcium-activated potassium currents, or, and that's basically the focus of our work, of my talk, via short-term synaptic plasticity. So it's sort of like indicating how many neurotransmitters can be released after an action potential. And the other key factor are basically fluctuations, and here I will focus on finite size fluctuations. Okay, so let's start with short-term synaptic plasticity. Like one of the seminal models by Zodix Markram was, if we have like a neuron, like here, that elicits spikes, then usually this presynaptic spike train is modulated by facilitation and depression variables that amount to like the number or the amount of neurotransmitters that can be released after that action potential. So whenever a neuron elicits like a spike train, what actually comes out or comes as the input to a postsynaptic cell is pictured down here. So you see if like neurons are firing, or like if the spikes come rapidly one after the other, then the effective spike or the effective spike train decreases. And then after a time of no spiking, then the spike strength, the input can be increased again. So this is one neuron. Let's have a look at what happens if we like simulate a full network of those neurons. So here we have like 100 neurons. There's no fluctuation. They are all coupled. They're only like, the only thing that's in there is they have like this short-term synaptic plasticity. And we see that there are like different states visible in this neuron. Like for instance here, like all of these neurons kind of spike together. So this is like a population spike. Then there's a period of silence in between, and again some other neural activity. So every dot in here means this is one spike of that neuron. And yeah, we have like exactly these population spikes, and sometimes, not after every population spike, but sometimes after such a population spike, we can enter in an up state. Like here we have like the population activity, or the mean firing rate of our population, where we see like we have an up state, and between population spikes, then we have down states. Another important factor here is like if we increase the network size from like 100 to 500 neurons, like this is this orange line, then basically the firing rate, the mean firing rate, the mean population activity remains silent, and there are no transitions between these metastable states. So the goal of this talk will be to derive a mesoscopic mean field model in order to better understand metastability that arises from the interplay of a slow fatigue mechanism, here short-term synaptic plasticity, and the finite size fluctuations inherent to our network. For simplicity, I only like look at short-term depression. So we only look at like the depression variable and forget about the facilitation variable. So here, H, just to guide you through the equations, H is, or HI is the input potential that every neuron I receives. There's a common input mu. Then we have like this leak term, tau is like the membrane time constant. And then we have here like an interaction, like an all-to-all interaction term, all-to-all coupling term, where we have xj's, which are basically the available synaptic resources at a presynaptic neuron. And sj is the spike train. It's like a delta spike train that you see here. And in general, we have like u naught, like a mean facilitation variable, times xj times sj, which is the modulated spike train that we saw before. So this is basically this U naught SI, what you see here, this modulated spike train, what comes into that. Okay, I also have to tell you, okay, this the SI is like a delta spike train, which occurs, or well, where every spike is modeled as like as a time homogeneous Poisson spike with intensity f of h. So this f of h is like a smooth threshold linear transfer function, and you can think about that as like some expected mean activity. All right. So how can we go from here, from this microscopic network model, where we consider like n neurons, to a mesoscopic network model describing like a mean field model? Importantly, in this mean field model, we have like to consider two sources of stochasticity that shape the fluctuations of our finite size network. So one is really the spiking output, like this shot noise stemming from the Poisson, like from this Poisson spike train. And the other one is that we have like heterogeneous depression due to the individual spiking. So it's like this xi variable, like the synaptic resources that will like differ over our whole network. So in order to describe our mesoscopic mean field model, we have to take into account both of these kind of sources of stochasticity, and they, as I will show, they will shape the fluctuations of our network. Okay, so like a general idea is we introduce mesoscopic variables h here and x, which basically are the means over the hi's and the xi's. So we can write that down. We end up with these sort of mean field dynamics, but the problem is that we have these sums in here that still depend on the individual neurons. And the basic idea to overcome these sums, to introduce them or to replace them by mesoscopic variables, is to make use of a chemical Langevin equation. So we have a look at this integral down here, and the time or like the increment during a macroscopic infinitesimal time step in the limit when we have like a lot of neurons. So this term here can be described by a Gaussian with mean and variance. And while usually heuristic macroscopic models forget about this this variance, we here make, well, explicitly account for the variance as well. We see the variance here scales with 1 over n. So we really have like these finite size fluctuations. And on top of that, we have like this factor q in here. Q is basically the second moment of all the xi's. And yeah, in order, like our mesoscopic mean field model will also describe the second moment of the xi's of the resources. So to spare you like a lot of the details of the derivation, in the end, we end up with this mesoscopic dynamics for our mean field h, mean field x, and for the second moment q. And as you see, like the only term where we have like the noise term is that this noise is basically shaped by q and also by f of h. So we have like some kind of multiplicative noise that shapes the dynamics. So interestingly, in this diffusion model, like a first step is we can go to the limit n to infinity. And if we take n to infinity, the noise vanishes and we end up with I call it with the macroscopic dynamics, or I call it this deterministic backbone. And usually this deterministic backbone can give us like hints at like all the dynamics that are relevant for the actual network behavior. So for instance, just looking at the deterministic backbone, like at this two-dimensional system, we can apply dynamical systems theory, draw the null clines in this hx plane. We can determine the stability of the fixed point. So in general, we have like here a stable fixed point. The input potential is quite low. The depression level is quite high. So this is a stable fixed point. Then we have like an unstable fixed point, here, and in between this diamond, this orange diamond is a saddle point. So we can also have a look at the saddle manifolds of our saddle point. The stable saddle manifold is the separatrix. And if we have a look at the unstable manifold, then it gives us the orange dotted curve. And this is interesting because if we really simulate our full mesoscopic model, then the stochastic neural trajectory will follow this unstable manifold. Once we are on top, above the red separatrix, we follow the unstable manifold and make this excursion back to our unique saddle point. But because of these finite size fluctuations, at some point we will be kicked beyond or kicked across the separatrix, and then run along the orange dotted curve along this unstable manifold back towards the fixed point, to this low activity fixed point. So if we look at a trajectory over time, then we see in this H dynamics of our input potential really these spikes, these population spikes, and the X variable goes like that. Okay, so here we have a nice population spike diffusion model. How does this compare to our microscopic network dynamics? So in order to compare the dynamics, we can run simulations for as few as 30 neurons in our network. Have a look at a qualitative look. This looks pretty nicely. And also if we look at the frequency of our dynamics, then we see a very good match. Also the interspike intervals, if we only look at the differences between these population spikes and compute the probability density function, then we see a very good fit between the microscopic and the mesoscopic model. There's a little bit of divergence here in the peak, but as soon as we increase the number of neurons, they go to 200, then we have an almost perfect fit. So the larger the network size, the better our diffusion approximation, the better our mesoscopic mean field model. Okay, now let's change slightly the parameters. So changing the parameters makes this upper fixed point to become stable. Before it was unstable. Now it's stable, and it lies inside an unstable limit cycle. So as before, whenever we have a trajectory starting close to this low activity state, then finite size fluctuations will kick us across the red separatrix. We make an excursion running along the unstable orange dotted manifold, saddle manifold, and on the way back to our down state, we come very close to the unstable limit cycle, which is the basin of attraction of our stable fixed point here. And finite size fluctuations can kick us inside it. We try to run into the upper stable fixed point until other finite size fluctuations will kick us out of the limit cycle, also out of the basin of attraction, so that we run back to our stable down state. So this is exactly this transition what we have here. From the down state, we go to the up state. We have this kind of ringing behavior, which, the frequency that's in here corresponds to the stable focus of our stable up point, and then we can switch back to the down state. So that is basically the dynamical insights, the dynamical mechanisms that is hidden behind our mesoscopic mean field model. And again, we can compare this dynamics, what we've seen in the mean field model, with the microscopic networks here for 100 neurons. Again, it looks like the qualitatively looks very good. Also quantitatively looks very nice. But there's slight deviations here, and especially if you look at the histogram of our potential, then we see that there are some stronger deviations that we had before. And but the nice thing here is that we can make up for these deviations because the discrepancy here that we see between our mesoscopic diffusion model and the microscopic network is that this discrepancy is due to the spiking output, this Poisson shot noise. And by going a step further and introducing like a hybrid model where we explicitly model spiking output as Poisson shot noise, we can get like a perfect fit. The dynamics will look like that. So here this dz dt is then the Poisson noise, but I don't want to bore you again with the details. Yeah, the result is that this hybrid model really captures network dynamics for arbitrarily small network sizes. So even here for n equals 30, we have like a perfect fit where before the diffusion model wasn't as strong as it was. So as an intermediate summary, yes, we can derive a mesoscopic mean field model for like spiking neurons with short-term synaptic plasticity. It's important that this is a bottom-up approach. And the accuracy of our diffusion model depends both on the network size, but also on the dynamical regime. So when we have like this transition between up and down dynamics, there it was important that we really concentrate or focus on the particular realization of our Poisson output. And as an add-on, this mesoscopic mean field model, this new mesoscopic mean field model, allows for new insights into hippocampal replay dynamics, and that's the part that I want to focus on the last couple of minutes.
You have 7 minutes.
Right. Okay. So, so for the replay dynamics of place cells and hippocampus, usually if we have like a rat running around a corridor, then it's known that we have like kind of neurons here in different colors that code for particular locations of our rat. Interestingly, once the rat kind of stopped running through this corridor and it starts dreaming about that, then we have like this replay. So without being in this exact corridor, the mouse or the rat like rethinks of this trajectory, and this is called replay. So we have like this kind of sequence of active patterns now going in reverse. Sometimes this can also happen in the reverse direction. This is called preplay. So this is a nice cartoon, but it also exists in real data. Usually people measure data from CA1 because it's easier. But yeah, to model that, it's better to like really look at hippocampus area 3 because there we know that we have some sort of recurrent connectivity, so that we can kind of store we can store an environment in the coupling of different neurons or different populations within the circuit. So one of the important models was by Romani and Sodix like 7 years ago, where they considered like a circular environment, like sketched down on the left. We have like, or they took like 100 heuristic mean field models, coupled them via like this Mexican head coupling functions. And what they found was with the short-term plasticity that you see kind of metastable activity here on the average activity, like averaged over all 100 populations. And from time to time, you see like these bursts. And even better, if you zoom into one of those bursts, then you see like that actually there's trajectories running across the place position, like the place field position. So you run into like either like forward or kind of backward direction. Then they did like a lot of statistics, but again, like for the sake of time, I try to skip that. So we try to model exactly the same kind of like hippocampal dynamics with our model. So instead of using Romani and Sodix's deterministic firing rate model, we used our mesoscopic description. So in the middle, we have like our macroscopic simulation, the left, the mesoscopic simulation. We also see like this kind of bursts. So there's a good fit between mesoscopic and microscopic simulations. It also fits to like Romani and Sodix's results. An important difference, however, is that in our case, it's a pure finite size effect. So as soon as we increase the number of neurons per population, like going n to 5,000, then we see like the zero line, no activity, no replay dynamics at all. From the statistics here, there's also a good fit. It looks qualitatively the same. So what are actually the differences? And the differences, yeah, just to tell you a little bit more. So if we look at the sequence of these kind of replay dynamics, so if we look at the sequences of replay dynamics, like sort of the intervals between two bursts, then in our model, in our mesoscopic description, here we have a kind of a unimodal Poisson-like interburst interval distribution, and there's hardly any serial correlation, so of like between consecutive forward or backward replays. Whereas in Romani and Sodik's deterministic model, you have like a bimodal distribution of these interburst intervals and also strong serial correlations. Comparing that to experimental data, like here are some examples, it looks like that experimental data really shows a trend that we have a unimodal distribution, which kind of like makes, well, supports our idea that it's kind of like a finite size effect. But it's also possible that like a combination of fatigue or like fatigue mechanisms and finite size effects like together form this metastable hippocampal dynamics. And as a last point to end is we also tested one of the other hallmarks of hippocampal replay dynamics, namely storing different environments in the recurrent connectivity matrix. And then you see that you have replay of just one environment. So here's like one environment, environment 1 is active, then environment 3 is active, whereas the other two are different. And the results basically are that in our finite size model, or in this finite sized induced regime, the replay dynamics are way more random than in deterministic models. And yeah, more information about that is in our archive manuscript. So yeah, thank you very much. And if you're interested, have a look at the archives.
Thank you, Bastian. So yeah, we have time for 1 or 2 questions. Please just either unmute yourself and ask a question directly, or write something in the chat. And at the moment, there's nothing in the chat.
I had a question. So can you hear me? Yes. Okay.
Who's speaking? Sorry. Okay.
It's me.
No,
Yeah, it's me.
yeah.
I didn't turn my video on. Sorry.
Okay.
Thank you for the great talk. I was wondering in the simulations that you had for the Mesoscopic model for the place cells, where you see this nice sort of spatial pattern. So do these come about then spontaneously from an like, what kind of stochastic input is put into the model so that these come about?
There's no stochastic input at all. So it's like really, well, the only stochasticity in there is that we have like stochastic firing, but there's like the external inputs are deterministic. It's all receive the same input. It's kind of like stationary. So there's nothing that triggers single events. Like all these bursts that you see here are kind of like really metastable states where where kind of like the finite size fluctuations of the network, well, it's not really finite size, it's kind of like high dimensional, right? I mean, like finite size fluctuations are like this very high dimensional dynamics well, leads to these kind of high activity states.
All right, that's very nice. So what was there's the same input to all the
same input to all neurons, no external noise. Exactly, it's all inherently generated.
And are those periodic inputs or?
No, it's like stationary.
Oh, okay. All right. That's pretty cool. Okay, thank you.
Okay, is there another question? Otherwise,
there are a few, I think.
Sorry, maybe I.
And one other person had their hands raised on the Zoom before.
Okay. No, sorry, I didn't see. So I see there's several. Excuse me. So Gabe, you have a question. So maybe a short one because we have also to move then or further.
Sure. Yeah, I had I guess one really quick question and one maybe slightly longer one. So the really quick question is that like 30 is remarkably small. So does the hybrid model, does it fail for 5 neurons or for 1?
Oh, good question. I didn't test 5. I didn't test 1. It's like it worked for 10 and 20, or for 20 it worked remarkably well. Like, and then the question is, why do you need it for 5 neurons? Then you can also.
Yeah. My second question was, so here you suggested that kind of the hippocampal replay was due to these kind of finite size induced network reactivations. And my understanding, at least, is right, is that you have fluctuations in these networks through finite size because of this 1/n coupling, right? Like without the 1/n coupling, if you had stronger coupling, right, then you might not lose fluctuations in the large n limit. Or you could also think about other kind of intrinsic sources of noise in these networks or variability, like stochastic vesicle release, right? Like the microscopic model that underlies this short-term depression. So do you have any thoughts on comparing those like different potential sources of fluctuations on the statistics of these reactivations?
Well, honestly, I have no idea. I haven't thought about that, but it's a really great question. It's like really, really interesting to see, okay, if you make these xi's and like here in those dynamics, if you make like these xi's like stochastic on top, whether that gives you like more dynamics. I mean, if you like just going from the microscope,
maybe you can discuss. Sorry if I break you. Maybe you can just discuss
sure.
later together, because we are a little bit late in time. So I would like to stop here and move to the next speaker. Sorry. So